from Microbe TV, this is TWiN, This Week in Neuroscience, episode 64, recorded on October 6, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today from New Orleans, Vivian Morrison.
Hey, guys.
Is it really hot down there?
No, actually, it just changed. Today's a little moist, but it's much cooler. Still hot by y'all standards, I'm sure.
Yeah. Are there seasons down in New Orleans?
Yeah, they are. It's just that they're
super hot.
Some of
and
them
muggy.
are shorter.
Okay.
Yeah.
Okay.
We, New York, we never really have fall. We have fake fall, and now we're in this fake fall where it's warm again. And then it's fall for one day before it turns to winter. Today it's 24°C and sunny. Also joining us from New York, Tim Chung.
Hello. Hi, Vincent. Hi, Vivian. I think I only know it's fall, or in the UK we say autumn, when the colors change to the leaves. That's my...
It's just starting to change here in New York, right?
Really? Oh, I was going to take a trip.
Yeah.
I was going to take a trip up to Boston, and I was like hoping I'd catch the tail end.
Oh, you have a couple of weeks.
Yeah.
October, most of October. Boston, when I was up in New Hampshire at the beginning of September, they were just starting to turn a little bit. Mm-hmm. It's okay. Fall colors. Okay, we are back. We missed a month, right? We missed September. It was my fault because I was traveling a lot. So we will make up for it today with an almost unintelligible... No, I'm just kidding. A very difficult paper. Vivian will make it intelligible because...
I'm going to try.
So take it away, Vivian.
Well, it's ironic that you say that. I'm going to help it make sense because I think when I shared this with you guys, I said, I'm going to attempt to discuss this paper. So if it's tricky for me, if it's tricky for Tim, and if it's practically unintelligible to Vincent, then I think it's fair to say that the time that we have today doesn't allow for a deep dissecting of some of the pieces of data and some of these findings. And I don't want to, I don't want to publish something that people or have us post something that people listen to the first few words and think, I have no idea, what the point is or so what I'd hope for us to do today is to hit the main points, focusing on what is the takeaway message. So maybe focusing focusing a little bit less on the methods. I know that sometimes the papers I present, I really lean into the methods, especially when it involves fluorescent tracers and genetically encoded tools to label cells or stuff like that. And so while we're going to be hitting on the main points, the main takeaways, and thinking about what are the interesting extrapolations, at the kind of gross level, what do people care about? But I also want to take some of this time to review some key elements of neuroscience that are necessary to make sense of this paper. And then we were discussing before that we may be able to put in a little bit more in the show notes for those of you who are interested in understanding some of these elements a little bit more deeply. So hopefully that's something that will come to fruition. And if you like that idea, please let us know in the comments because that would be really helpful to gauge interest in going deeper. But we'll start off with the paper now. And as these kind of basic or more fundamental, not less complex, but more fundamental aspects of neuroscience that we need to understand come up, we'll dive into those. So the basic question, if we're going to take a 15,000 foot view, is that we have limited real estate in our brains, and we have a very rich visual environment. And sorry, I think my phone just buzzed. And we have to make sense of all of the visual stimuli that we receive, because it's not like you can just like turn off one eye or turn off half of your field of vision and only see or receive input from one half or one eye. We have to take it all in. Yeah, or you have a stroke, or or multiple sclerosis. So, but that's not intentional, right? That's like that part of your visual field is out of order, under repairs. But we have all of this stuff coming in, and we're going to talk a little bit about the visual system and how it takes all of this stuff in and builds it up so that we can understand the world around us. But we have essentially how it works is we recognize lots of tiny little pieces of data, if you will, lines that are high contrast, so black and white, or basically just bright and dark. And we can see different colors, obviously. It's not like the information comes in and is just black and white. And then we have these kind of increasingly large domains of neurons that help piece all of those little lines together to create a picture of what it is we're looking at. And then through a kind of like black box magic-y thing that takes, years and years several years during childhood to develop, we're able to connect that visual picture to a concept and understand what it is and how it fits into, what we're supposed to do with it. Are we supposed to pay attention to it? Are we not supposed to pay attention to it? How important is it going to be for changing our behaviors? But like I said, we only have like this much room. How do we code everything? How do we fit it all in there and do all of that processing and building? And so that's a really, I think, fascinating question. And it's one that I would say maybe next to the auditory system, which is a little bit different, but it does also have, all of your senses pretty much have a map. Although I'm not sure that, does the olfactory system have a map? I don't know if it does. Like,
no idea.
Physical map, you mean? Like
a somatotopic map? We have a map on our tongue. It's also like the somatosensory map. We have the homunculus, which is like the map of the little man that goes, it's kind of maintained all the way from the spinal cord up to the cortex. We have a visual field. So on the retina, there's a map, and that map is maintained. That positional information is maintained as you go from the retina all the way up to these higher processing centers, or even ones that are outside the visual system, but that will put together, for example, auditory and visual information and understand more complex situations. And then with respect
there certainly is an olfactory map. I just looked it up.
Oh, okay.
Right, different odors mapped to the olfactory epithelium. Yeah.
Right. Okay. Yeah. Yeah. So in any case, it's interesting I forgot why I was talking about maps, but it's a fascinating topic. That's why we're talking about it today. So I want to start like a little, like I said, down at that very basic level. I'm talking about the visual system and its organization, and I'm going to rely on Tim a little bit to like hold my hand through this, because I'm sure you learned some stuff about the visual system.
Dig deep in the memory.
Yeah, me too. We're going to both dig deep. Right. And so basically, like, I'm not going to get all the specifics right or even get close to them. But like I mentioned, you have a retina, your two retina, and they correspond to different parts of the visual field. So if you like put them in, into quadrants, you have like an upper visual field, the upper visual field, the lower visual field, and then the right and the left. But as this happens several times in numerous systems, the left visual field actually corresponds to the right side of your retina. The right side of the visual field lands on the left side of your retina. And then your brain has to... exactly. And so then the brain has kind of routes them a couple times to get the visual information where it needs to be in the visual cortex. And so that then we have, like I mentioned, the small pieces of information are these basically contrast, information about contrast. And we have like individual cells, or very small individual cells that are very sensitive to like lines in different directions. So like high contrast. And I think they showed this, and it's something we could in cats.
Those are the cells that are very early in the visual processing system. So like the cells from your retina goes to this bit in your thalamus called the LGN, the lateral geniculate nucleus. And that is where I think the Nobel Prize winner Thorsten and Wiesel recorded from a long time ago, where they had a cat and then just showed it. They recorded from these neurons in the thalamus, and then they showed it like pattern stripes, just single line going through on like an overhead projector. This is like in the 50s, so they didn't even have computers back then. They had to show like old school overhead projectors. And then when there's a line that crosses the cat's visual field, going in one direction, but not in the opposite. Going in one direction, these cells would fire, but not if it goes in the opposite direction. And then there are other cells that are sensitive to maybe a line, just a presence of a line. But if you flip the line 90 degrees, those cells would become quiet. So there are all these very simple visual motif that these cells seem to be sensitive to. And then I think, I'm probably 80% wrong about this, but they then move to the next stop in the visual pipeline, which is going from the LGN in the thalamus to the primary visual cortex, the V1, the thing that is at the back of your head. And then they also see something very similar, where there are cells in your visual cortex that are sensitive to these very, very simple motif. So lines of different contrast, patterns of different contrast, lines moving one way or the other, like all these things. There are also like, there are sometimes cells that are sensitive if you have a dot in the middle surrounded by bright surround. And then, but if it is just if it is just a huge dot, then the cells stop firing. So there's like all these very basic elements that get stitched together. And the idea is that the primary visual cortex, the V1, the cells in there are sensitive to these very basic elements, and then they send their output to the next layer of the visual cortex system. The next region of visual cortex, which is the V2, the secondary visual cortex, I guess.
What's the spatial arrangement of V1 and V2? They're not layers, right?
No, very good point. They're not layers. So each visual region, visual cortex, it has 6 layers. So the cortex has its own layers, kind of like a layered cake. So those layers have a very well-defined motif. They're called cortical columns. They are organized in individual columns called cortical columns. And we won't go into all the details of which layer talks to which layer, because that's too complicated.
It's complicated. But this column organization is something you see all along the cortex.
Right,
everywhere.
Right, exactly.
So, but I think it's a little bit harder to map I think it's a lot easier to map those columns and layers functionally in the visual nervous system than in other things that are.
It's very well looked at because of these things called ocular dominance columns that we talked about a long time ago, where you can find a whole column that are sensitive just to your left eye or your right eye, and then if you do an experiment where the animal don't get visual input from one eye, if you put an eye patch for like when the animal is a baby, then those ocular dominance columns get remapped because there's not enough visual input and it's very plastic. So Anyway, that's a different story. But very quickly, all these tiny motifs in V1, they get stitched together. They get passed along to like V2, the next stop in your cortex, and then they start building up. You start kind of stitching these. The theory is that you start stitching these lines together. Every time you go to the next region, you pass to the next region from V2 through to V3 or V4, and then further down. Eventually, you go to like the temporal lobe, kind of a higher processing area where the cells would be sensitive to not just single lines, but a whole face. So these are like the famous Jennifer Aniston cells that people found a long time ago, where they recorded from the hippocampus or maybe temporal lobe somewhere, and they found cells that are sensitive only to like Jennifer Aniston, because presumably it stitches up all these kind of tiny little motifs together. And interestingly, that's kind of how neural network kind of works as well for computer vision. So it's kind of interesting. Sorry, very big digression, but that's a...
No, I mean, this is necessary, right? Then something that goes along with what Tim was talking about is the idea of the receptive field. So as you're going increasingly higher in increasingly further in the journey of the visual information, you'll have groups of cells that respond. Like you'll have it, like in this paper, they're like, there are two regions of the V2 visual cortex that they respond to smaller regions in V1, and then like V3 would include those, like one of them and something else, and then V4 includes all of them. So it's getting the receptive field is getting larger and larger. But with this also, it then begs the question, like I said, how do we maintain resolution of what we see? How do we prevent all of this incoming stimulation from being like mixed up and confused, and so that we can't make sense of the world? And one of the ways that we think this happens is by the precise, I'm not going to use the right words because I'm not a physicist and haven't studied brain waves, but is by the synchronization, or like the way in which different brain waves interact. So, for example, I know that, and we may have talked about this on a previous podcast, where there will be a certain kind of brain wave generated from the hippocampus, and it has to meet with a certain kind of brain wave that's happening in the thalamus at a very specific time in either one or both of their phases, and that generates a different kind of wave that has to arrive at the precise moment that another brain wave in the cortex is going along, and it's the timing and location of that wave, that lower brain's wave arrival in the cycle of the cortical brain wave that like delivers information. So if you don't have all of these waves lining up, then information gets lost. Like you cannot do memory, for example, or like take the kind of short-term, mid-term memories that are generated in the hippocampus and then like store them somewhere or somehow in the cortex.
Yeah, maybe I can have a very quick rundown of brainwaves, like a quick background.
Please do.
Because I remember presenting a paper a while ago on brainwaves and depression. Brainwaves were like discovered a very long time ago, like 100 years ago, when some dude, Someone, I've forgotten what his name is.
We'll add his name in the show notes.
In the show notes.
Sure.
So I believe some German, which is apt because the paper we are going to talk about today is from Germany. A German scientist was recording basically you can put electrode on the skin, on the surface of the skin, on your scalp, and you can record these basically voltage changes. And what he found is that whenever he does this to someone and that person is like daydreaming or like half asleep, he sees these. Well, if the person is alert, you don't really see much. You just see like kind of noisy stuff. But then whenever this person is like daydreaming and half asleep, you see this huge slow wave that he calls alpha wave. It kind of starts popping up. And this is like the birth of brainwave. And then people since then have discovered if people are half asleep or under certain anesthetics, you see these huge slow wave alpha waves. Whereas if the person is preparing for movement or is very paying high attention to something, you see these very fast, much higher frequency waves called gamma waves. And since I study Parkinson's disease, this is quite a big thing in Parkinson's disease, which is this in the middle frequency range called beta wave. You see a lot of it as the person is preparing to move in the motor cortex. Whereas in Parkinson's disease, they seem to have a lot more of these beta wave, where they don't transition from beta wave to actual movement. They seem to get stuck there. So all these waves are, very importantly, they're like correlates of what might be going on inside your brain. But in so much that I think I saw like a Wall Street Journal video on YouTube where they're describing people like grade school children in China have to wear these headbands that measure the brain waves to see if they are daydreaming. And if they're daydreaming, the app sends like an alert to the parents. So that happens.
Actually, that's super interesting. Could actually be useful.
I'm surprised it didn't instantly just shock them. Just zap them.
Yeah, I was actually expecting to like.
Anyway, I think
everybody daydreams.
Yeah, exactly.
Some of us more than others, though. If you think about the inattentive form of ADHD, it's a lot.
Yeah, I understand.
We can even.
But they're good. I would guess that every kid in the class at one time or another daydreams.
Yeah, but we can also argue daydreaming is actually functional. It helps memory consolidation.
Yeah, of course.
But that's for a different podcast. But importantly, these brain waves, there's been a huge argument as to are we just picking up this kind of side effect noises of, like, for example, your computer, and it is like, if you look at the computer screen, it's like rendering all these pixels to display for you. If you stick like a antenna next to it, you see this huge wave going up and down as it's rendering all these pixels, and you can actually use that to spy on people. It's this thing called von Eck freaking. Never mind. I got it. I was reading about this in a sci-fi book. That's why I know about this.
Was it Neil Stephenson?
It is.
Was it
Cryptonomicon?
You guys all know about this.
Yeah, that's right. That's right. Freaking.
Yeah. So, but anyway,
and you have to wear a Faraday cage.
Exactly. You have to wear the tinfoil hat or something.
The tinfoil.
Anyway, but if you stick an antenna next to your GPU, you see all these waves. But those waves aren't doing anything. It's just a side effect of your GPU displaying things onto your screen. But your brain waves in your head, like all these alpha waves or these gamma waves, are they just side effect that the antenna, the electrode that they put on your scalp, is it just a side effect they're picking up, or are they actually important for functioning? And this is what Vivian's going to talk to us about, especially for the gamma wave, which is very important which seems to turn on when you're paying attention. But a final thing I want to quickly say is that people have found they have previously found that, for example, if you're paying attention to an object in your visual field, then gamma wave kind of switches on. You see a stronger gamma wave component in your visual cortex. That is thing one. Thing two is that if you measure different bits... So, it's barely Dr. Tim. If you measure two different regions, when you're paying attention, not only do the two regions gamma wave turns on higher, they also synchronize. Because do you remember, a wave has peaks and troughs. And often you have these waves turn on and off by themselves, kind of spontaneously. They don't really synchronize. But when you're paying attention, they start synchronizing both within a region and also across regions. And people are debating whether those are important things, or are we just picking up the GPU kind of effect.
Yeah.
Anyway, I'm done.
I think that's perfect. That's really, really important also because there's the different kinds of brain waves, like the alpha, beta, gamma, delta, theta, that and they all have different frequencies, right? And where you look at the appearance of some of those waves in different brain regions and how the brain waves from those brain regions synchronize. But it's also, you might have, I think people might be like, oh, synchrony, like they're both moving like this. Synchrony exists in other forms. It just means that or coherence exists in different forms. It could just be that the two waves are phase locked, which means that like at every peak of this gamma wave, the gamma wave from somewhere else is going to be peaking at like 36 degrees from the peak of the original one. And so if you think about all of the combinations that you can have, if like all these different frequencies of waves being phase locked in very specific times, and that this could be even like could change as a function of the region or the task, it just kind of blows your mind, no pun intended. It's like how many different combinations of things the brain can do, and then how we can utilize all of those to make sense of the world. And this might be a good time to mention that the name of the paper is Gamma Band Synchronization Between Neurons in the Visual Cortex is Causal for Effective Information Processing and Behavior. So the information processing we've kind of talked about globally, and then the behavior that they're talking about here is nothing fancy. Well, it's kind of fancy, but nothing like too much based on like big movements. It's based on attention, and it's done in monkeys, and we can talk more about the paradigm or like what the animals are being asked to do, but it does involve them like moving a lever to indicate the re-arrival of something. So let me
tell us the authors.
Yes, sorry, I got excited about the behavior component. Eric Drebitz and is the first author, and there's only 3 authors, so we'll say all of their names. Lucas Paul Rausch and Andreas K. Kreitzer are there. I'm taking German on Duolingo, which is why I felt compelled. Although a German speaker would tell you I sound like a French person speaking German. But so the task that they're asking, I'm going to walk you guys through the task, and then we'll talk about how they're going to manipulate it. The task that they have the animal do is they have a shape, a certain part of the visual field that they can map where that spot in the visual field lands, so to speak, on the brain. So they're like, okay, this is the brain region that we need to manipulate later on. And the animal is going to be fixating on that area, and it's going to see shapes, and it's a shape that morphs over time. And there are 4 different states that it can morph into. So think of like a circle, then becoming a trapezoid, then becoming a bean shape, and then becoming something else, a cylinder. I don't know. But it's going to morph fluidly between these different shapes. And then the goal is for the monkey to, push the lever when it sees the original form reappear. And there's a very specific window of time following the reappearance of this shape that the monkey can respond in. And so they're interested in knowing how many times does the monkey get this right, and how long does it take them to respond. So reaction time, and then like wins or misses. And they have implanted a small electrode in the visual field, or the region of the visual field, or the visual cortex that corresponds to this visual field, and they're going to introduce a very small pulse of electricity and see how that affects. So they do it in V2, and they want to see how does this pulse of electricity in V2 change how the information from V2 arrives at V4. So I haven't thought of an analogy for this, but if there's a specific place along a brainwave that's occurring in V4 that is if there's a specific place on that wave where it's necessary for a preceding brainwave from V2 to arrive in order to control the animal's ability to focus enough on the original form to then say later, oh, it has come back and respond within that short time window. What happens if we kind of nudge the V2 input so that it arrives at a slightly different place? And does that mean that the animal is going to be less successful at identifying when there's that reappearance of the original shape? And how long is it going to take the animal to respond? I think that's the basic. But Tim, what else do you think is important?
I was just going to butt in and add that. So V2, as the number goes up, the more the deeper it is in the visual processing kind of relay station. So the V1 is the first bit of the cortex that receives visual input from the thalamus, and then V1 talks to V2, and then V2 apparently splits, and half of it talks to V3 and the other half talks to V4. Maybe it's not 50-50, but like some of it talks to V3, some of it talks to V4. This split is called the dorsal versus ventral stream. V3 is in the dorsal stream, is supposed to be important for movement-related visual information. V4 is the ventral stream, is supposed to be important for object recognition. So they're looking at V4, which is like a more object-type kind of processing pathway. But what's interesting is that V4 is the first the first visual region that if you put an object inside, so if you record from a neuron in V4 and you start mapping out where the receptive field is, you put something in the receptive field of V4, the firing rate changes if that object is being attended to. So for example, if you train an animal to pay attention to triangles but ignore circle, then in V4, if you put a triangle inside the receptive field of this neuron you're recording from, it would fire a lot. But if you put a circle that the animal has been trained to learn that it doesn't give any reward, the firing rate would barely get modulated. So V4 is very interesting because it's attention modulated area. Whereas V2, just one region upstream, it doesn't matter whether you put a triangle or a circle in the visual in the receptive field, the neuron would fire similarly. So some magic is being done in V4. So this is one of the things that they're trying to understand is how does this attention is modulating how the animal is how the neuron is responding to whatever input is from the visual field.
Yeah, and what you've just said makes you've mentioned the word, you brought up the word train multiple times. Because they're training the animal to say, like, this is the thing you're looking for. This is a distraction, which that kind of learning association doesn't happen in the visual cortex. It's happening elsewhere in the brain, right?
Maybe.
I mean, obviously, like, there's information. I'm just wondering to what extent there's going to be this like top-down influence of V4 activity. Or like the V4 activity and activity of the neurons in the V2 derived populations. To what extent is there going to be top-down regulation of that? Because like if you think of this is totally could be a useless foray, but the Wisconsin card sorting task where you have to. Or the what's the one? The Simons task?
Is that for attention set shifting?
Yes, it is.
Okay.
So you have these like on a really short time scale, your brain is you have to change what you need to pay attention to. And I guess but perhaps in that situation, you don't have any change of where the object is necessarily in the visual field, like physical location, which would have an impact on which cells are firing. But
not necessarily, but it would apply to the whole, your entire V4, presumably. Like if you suddenly have to pay attention from like the shape of something to the color of something,
which is part of that Wisconsin cards task. Like what's the rule?
I'm not 100% sure how that would be predicted to affect V4, but you would expect that might have an effect. But not in V1, for example. Like V1 and V2 might just recognize like the simpler, basic visual motifs.
Yeah, I would agree with that. Anyway, that was an interesting open-ended question. So they're going to go ahead and do introduce this electrical pulse into the V2 areas. And they do as good scientists should. They're introducing these pulses randomly, but within a certain window after each morph cycle. So each time the shape goes from being a circle to a square, for example, it's not their shapes are much more complicated. But from a circle to a square, like at the end of that morph cycle, or maybe it's at the second half of the morph cycle. So as it's approaching the next state is when they introduce the intracortical microstimulation. And I'm not going to go into it because it's a lot for me to keep in mind, but they're keeping track of where these things are happening, like where in terms of like how the task unfolds. They're introducing the stimulation. They're taking into account how long that stimulation is and how that might affect the timing of the neurons that are working in that particular part of the visual cortex. And they have to do all of this to kind of rule out certain other possible interpretations of how the brain does this. But like I said, I'm just mentioning it as being like, that's good that they did that, because they were taking into account that, like, yes, if this arrived at this time, or if effect A happened more when the stimulation was put at within X milliseconds or 0.05 milliseconds of this other thing, then this is the result we get. And that could potentially be a confounding variable when we go to interpret our main findings.
Sure.
So I thought that was admirable. Or maybe it's always it's necessary.
Yeah, I'll just kind of expand on this a little bit. So what the authors do, Vivian already mentioned already, but they are stimulating V2 and they're recording from V4. So V4 is the interesting part that is attention modulated. And the hypothesis that the authors have is that the gamma brainwave, the gamma oscillation in V4, is very, very important for. It's causally linked. It's in the title. It's very important. It actually allows the monkey. So these are recording in monkeys. In case we haven't made it, we haven't spelled that out. Allow the monkeys to perform the task. And so these electrodes have the very important task of actually recording both the neurons firing and also the brainwave of the surrounding region. And the idea is that as the trial starts, presumably the monkey is paying attention. And as soon as the monkey is paying attention, these gamma waves start going up and down, they switch on. And the task is designed such that the microstimulation from V2 come kind of at random time in the gamma oscillation cycle, like exactly which phase of the cycle it is in. The design is such that it comes at random time. And then the experimenter is going to collect all these data of, they will have some data when the stimulation comes at 0 degree, some data when the oscillation comes at 90 degrees. So like imagine like a sine wave, you have different degree telling you where you are along the sine wave curve. So they basically have data for the entire sine wave, where in the phase it is. And then they can analyze which, when they stimulate the V2, which bit of the gamma phase is important for the mouse to do things. And that is how you tell apart that gamma might be very important.
Yeah. Mm-hmm. Right. So and they actually show, they have a graph that shows you where the ineffective phases of the gamma cycle are in V4, and then what is the effective zone. Because it's there might be like a point along the gamma cycle where you're going to get like the most bang for your buck. Essentially, you're going to be able to disrupt the or alter the synchronization or the activity of V4 neurons the most if you do the microstimulation at this point. But as you move away from that point, it doesn't mean that like there's no effect. There is still an effect, but they use the word like decay to convey that it's less and less strong. So that's actually really cool that they're able to show that the changes that they may or may not see in terms of wins or misses, and also the reaction time, the degree of those changes are a function of the degree of changes in V4 activity. Wow, excuse me.
Okay, but just to, as Vivian's is recovering. But yeah, the researchers basically found, so I guess the headline result is that, as you might expect, if you go around going around randomly stimulating your visual cortex, it's gonna maybe negatively affect your task performance. So they found that if they stimulate V2 as the monkey is focusing and trying to perform this visual task, they get worse. In that they miss, they sometimes completely miss when the shape, remember the shape is morphing kind of continuously, and they have to, within the time window, tell they press a button when the shape goes back to the original shape. If you stimulate V2, they miss a lot more of the time. Not only that, when they don't miss, they also take longer. So the reaction time is also longer. So that is
would there be a situation with us that would mimic that without any experimental? Is there something in real life that would mimic that? I would. So they actually talk about this. I'll talk about this. They talk about this in the discussion. But you know how in cartoons, when I don't know, Donald Duck takes a great fall and bangs his head, he starts seeing stars spinning around. Mm-hmm. So what actually happens is, if you get a concussion, you can bang. Often is banging the back of your head, and that is where the visual cortex lives exactly. And you do see like you see bright spots flashing in and out.
I learned in the paper is called phosphenes.
They're called phosphenes. I'm sure it has something to do with.
You can get it from rubbing your eyes. Yeah.
Anyway. Oh, if you press hard on your eyes, you get those.
And it's also weirdness. Like in migraines.
Yeah, and it has something to do with your visual cortical neurons firing in a way that doesn't have anything to do with visual input. So like if they're firing willy-nilly, you start seeing spots. So those you would expect, if that happens very severely, you're not going to be able to, it's going to distract you from your visual task.
Yeah, sure.
So that is actually a confound that they talk about in the discussion, and they don't think that explains the data. Because to elicit phosphenes, to elicit these stars that the cartoon character sees, you need a much, much stronger electrical stimulus. So they have tuned their stimulation very, very well so that it gives the tiniest nudge possible to have an effect.
Yeah, they say that the pulse trains that would be needed to generate the stars are somewhere between 100 and 300 hertz.
which is much less than what they're putting in, and it would have to be for the hundreds of milliseconds. So it would be significantly longer. I wanted to just share a personal story, a personal experience. Several episodes ago, it must have been before September 2024, because I was pregnant when I presented this paper. It was about the changes that occur in the brain during pregnancy and the early postpartum period. And they mentioned that there were changes in something called the default mode network, which I think is fascinating. But the default mode network is basically that there are certain areas of the brain that are turned on. Really, they're turned on when the person is daydreaming, but you can't really tell, like, externally if what the person is doing when they're daydreaming. So it's really like the regions that are involved in the default mode network are characterized by the fact that they are quieted when you are involved in some, cognitively engaging task, like trying to pay attention to something. For, like we mentioned, people who, everybody daydreams, but some people daydream like more frequently or more hardcore than others. And I have actually had the experience, and I'd be interested to know if anyone else has. Because
just picturing in my mind a hardcore daydreamer, committed hardcore daydreamer, extreme like daydreamer.
Yeah. I think I'm going to make a T-shirt, extreme daydreamer. But we can add it as like the
drinking
TWiN
energy,
swag.
Drinking Monster energy drink and daydreaming.
Well, that might make it worse, You never know. But that there are times when I'm so deeply involved in my daydream that my visual field goes white, and I have to come back. And I'm not having a stroke, as far as I know. I'm not having a seizure.
Is it a vasculature thing? Like, you're not standing up very rapidly.
Right. It's not the same thing. But what you just brought up is that a lot of people, a lot of people now know about the orthostatic hypotension.
Hypotension, yeah.
Yeah, which is when you, when you stand up too quickly.
And your BP drop.
But that would be a blackout, not a whiteout.
Actually, that sometimes happens to me, and it is not a blackout for me. Oh. It's a whiteout. Okay. And it's like, then it's as if, if you've blown hot air onto a cold window, and then it kind of, But anyway, I think it's interesting, and it makes me wonder to what extent the activation of the default mode network kind of hijacks whatever incoming information and the coherence and synchrony of the that we're talking about here. How that that kind of that interplay. So something for people to muse about, or maybe dig deeper into the literature to see what other pieces of information might help explain that. But yes, they show that like Tim was mentioning, they offer reasons as to why that phenomenon would not account for the increased number of misses or the increased reaction time when they do win. And then they also ruled out the possibility that the misses and reaction time have to do with eye movements away from the focal point that they're showing the monkeys.
Yeah, because you can imagine, like, if you stimulate V2 and the monkey maybe see something in its visual field and it gaze over there, and then it would miss whatever it's supposed to pay attention to. So the researchers track the eyes to make sure the eyes weren't moving when they're stimulating. So,
which reminds me of something else. I'll have to look it up. But so on the retina, like we said, that there's a map, and there are different kinds of cells on the retina. So that vary in their sensitivity to light and how they get turned on or turned off. So we have stuff right in the middle, which is if you're focusing on something, you're giving a lot of stimulation to the very center of your eye. And so those have a special kind of cell. And then as you move to the like periphery, you have a slightly different type of cell, and those work better in low light conditions.
Right,
and the
those
ones
are the
that are
rods.
in the center, right? And then the cones in the center of the eye are good, and they respond well in like bright illumination, which is why you might experience this when you're like in a dark room and you can't see something. Don't look right at it, because the cells there will not be able to. They're not going to fire. So if you look at it using your peripheral vision, you actually will be able to resolve what it is. And then relatedly, if you move your head sometimes like this, then it can piece together. I'm not saying you should just go like this and expect something to happen, but sometimes you can your brain can see something better if you move your head. So taking advantage of that
like
dorsal ventral divide. Yes. And when if it's something in the corner of your eye or something you can't focus on, like that's moving rapidly or moving through something like trees or a fence, moving your head will actually help you see it. So you should, not that I think people should go stand next to microwaves, but next time you're heating up your lunch or whatever, go up to the microwave and try to watch whatever it is that's going around. And I would bet that you're not going to be able to get like a real good sense of like what's going on in there. But if you go like this and move, because it has like a grill kind of like
with the dots in it.
With the dots in it. If you go like this, you will be able to see through it much better and actually see what's inside.
Oh, yeah, maybe. Because I notice when I walk up to it, I can see. Then when I stop,
isn't that fascinating?
We are all T-Rexes. It is true. We are all T-Rexes.
Yeah, of course.
Yeah.
Neuroscience is amazing.
Yeah, it's really cool to experience things like that and be like, oh, that's my, what did you say? It was dorsal is movement.
This dorsal stream, remember. Perhaps, yeah.
But do you think if you are, let's say you're reading something and you're so focused on it, you're not going to see peripheral stuff going on? Would that be an example also?
I think that's a high.
I would say yes, and I would. Have you guys seen that advert, or maybe it's like a psychologist test, where the guy asks, there's a bunch of people passing around like a few basketballs, and then your task is to count how many times they pass a basketball to each other, and this goes on for like 30 seconds. And then at the end, they're like, okay, how many times do you pass a basketball? And people like tell them what the number is. And then the questioner says, did you notice the guy in a gorilla suit that walked through the screen? And most people do not notice it. And it's because you're focusing so much on the basketball. So this tells you about how strong like visual attention, like your V4 attention spotlight is really, really strong, because you just ignore everything else.
And that, I think, makes it, always looping back to what listeners can, what joy they might find from thinking about these things, is that that this isn't like a black and white thing. It's not like everybody performs that way,
Sure.
And if people perform differently, then, as they're showing in this paper, like the animal's behavior is an observable manifestation of like changes, electrical changes, and manipulations in certain brain regions, and that they can do this they can do this in a reproducible way. Although, side note, they only did it with 2 animals, but that's hard when you do primate research. And I was curious why one of the animals had almost double the number of trials as the other animal. Don't know what that means. I have a friend, coming back to the ADHD thing, but it's got it's about attention, so I have to talk about this. I have a friend who has ADHD. I don't know what kind. I have more inattentive, and we'll be walking. We were walking over a bridge in City Park, which is like a huge park, real close to where I live, and it has like a bayou in it. And we were walking along, and it's a very like rich environment. Like there's people bicycling, there's all sorts of trees. There's like, if you're on a bridge, which has like, wire and, wood slats and stuff. And then in the water below, there's lots of like algae and debris from trees and And we were walking along it, and I just went, there's an alligator in there. And she was like, what? And I was like, I just saw it. Like, I wasn't focused on the water, but I just saw it. And I had the same experience as a kid when we were driving through a rainforest on a family trip, like driving in a car, and I was staring out the window and was like, sloth. There's a sloth in that tree. And it like wasn't even close. So I'm like, this is fascinating. Not duh. We don't all have the same, attention or abilities to control our attention. And some of us are more in touch with our peripheral vision than others, and can actually appear to be focusing on something, but attending completely to what is in the peripheral visual field. So I go home, and your homework is to be having a conversation with someone and looking at them in the eye, and then shift your attention to whatever's happening over here.
I suppose that's a necessary skill if you have 3 kids.
Oh, it is. Like, my husband will be like, where's the baby? Are you watching him? And I'm like, I am literally I can see him out of the corner of my eye. I have a sense of where he has moved, in the physical world, and I'm always listening, So yeah, that's true. That's another interesting idea. I wonder there's lots of interesting questions, but the takeaway from this paper, because I know we've talked a lot about important things and made some gone on some tangents that are fun or not to listen to, but the takeaway message is it does matter. It does matter where V2 incoming information arrives in terms of the gamma oscillatory activity of V4. So if you disrupt, or if you change the arrival time of that V2 information, then it increases the reaction time when they do identify the return of the original shape, and it also impacts the animal's ability to even get it right. So to stay focused and to ignore a distractor. Expand, Tim.
Yeah, I was just going to, that really is the whole point of the paper. It's not just that when you stimulate V2, you disrupt the task, which maybe is not super exciting. But in the title, mentioned in the title, the authors really want to see whether the gamma oscillation plays a role. So what they did was they actually analyzed, because they had a record of where the oscillation phases when they stimulated the V2, and the stimulation V2 time is random. And then when they align everything together, as Vivian said, the effect of V2 stimulation consistently is way more effective, 30 times more effective. I lied. Twice as effective. Around twice, maybe. Yeah, maybe twice. I think maybe twice as effective when it is in the most effective phase of gamma, when it all lines up. So maybe I'm putting words in the author's mouth, but the one way of interpreting this is the gamma oscillation kind of acts as a filter, an on-off switch of when it allows information from V2 to come through. So when the gamma is at a certain, let's say is at the peak phase, then V2 information can come in. But when the gamma is at a trough phase, let's say, then V2 is ignored, and maybe V4 itself is doing its own stuff. So they can focus on doing its own stuff. So this is one way of interpreting what the gamma is important. What they found is that definitely the effect of stimulating V2 to disrupt this task depend critically on the gamma phase. Mm-hmm. So, and that's why they think gamma is causal for this particular task.
Is it fair to say that one takeaway message for non-scientists would be that not all visual signals are treated equally?
Not all visual signals treated equally, and also the same visual signal at different times is not treated equally. So it's also very timing dependent. And
do you mean in a in like a big way, like
across millisecond, based on the gamma phase?
Across milliseconds, yeah.
Like,
but also probably at a minute, from a non kind of electrophysiological point, like what's going to decide what is important or not important.
Completely correct.
But the same object, the same visual information is going to be salient in one condition and then useless in another. So it's not the actual, like, it's not the properties of the thing that you see that determines whether or not it's salient. It's much more than that, which is why I was talking about like the top-down modulation of V4 activity is of interest. But anyway, I interrupted you, Tim.
Yeah, it's really interesting because I thought everything your eye picks up means something. It's all important.
Some of us do feel that way.
but think, like, for example, like a stop sign, before and after you learn how to drive, it means completely different thing. Like, especially if you miss a stop sign, it's like $100 fine. So like things like that, you learn to have certain different significance for sure. But yeah, they mentioned
red, yellow, green light.
Exactly.
Yeah.
Mm-hmm. Exactly. But what the paper is interested in is really this timing relative to gamma phase. And gamma is a very, very fast oscillation. I think it's like 40 hertz to maybe 100 hertz. And if you
30 to 100 is what they say.
So if you turn it into like how many millisecond, one cycle of gamma is maybe around 15 millisecond. So it's very, very short. So you can fit 40 of these in a second. And if what these researchers have found holds, and if you can generalize, then they mention in the discussion, it has really strong implication on things like if you do brain-computer interface. Do you remember we talked a bit about we talked about using the brain to control your limbs and stuff like that, but
right.
Also, people have been thinking about like Neuralink and all these other brain-computer interface for people with ALS and other kind of motor disorder that you want to bypass your muscles and go straight from your thinking to maybe typing, controlling a mouse cursor, moving around. When you stimulate the brain, might strongly depend, like how effective it is might strongly depend on the gamma phase. And if that's true, then it might mean we have to redesign, like the designing of these brain-computer interface might need to like really pay attention to things like this. So it is actually quite not just kind of theoretically interesting about what gamma might be doing as a filter, as an on-off switch, but also for practical implication. It's quite important.
And these kinds of phenomena exist for all of the brainwaves, and for or maybe it's possible that some are going to be like weighted more heavily in terms of organizing certain kinds of behavior. But like beta oscillations and their timing will have impacts on other things like synaptic plasticity. So it's really like there's a lot of open questions, a lot of areas to research.
But one thing that might be worth mentioning is the reason why these brain waves might end up being important is because these brain waves don't exist in a vacuum. They didn't come out from nothing. They actually come from the mechanism where you detect them because you've picked up these oscillations because the brain is firing in a synchronous manner. So it is actually doing something important when these brain waves arrive, and you're just picking up a little bit on the signature. So kind of like the GPU, you're spying on your GPU. As it turns out, the wave from the GPU is very important, but the wave itself is not important. It tells you a little bit about what the GPU is doing. So maybe the reason why these gamma phases is important because the underlying like synaptic transmission is very important during that time. It tells you a little bit something about that. But exactly what is the synaptic mechanism for these brainwaves is still really hotly debated as far as I can tell.
Yeah, I mean, you're moving along orders of magnitude. I think it's of size and electrical activity. So it is hard to kind of find where those things converge and where those conversion points might differ in different contexts.
memory retrieval, for example, versus attention.
I bet this changes with aging also.
Oh, interesting. So
I was also wondering about it on the other end, that
when we're
like
younger,
for young brains, yeah.
So Benjamin Button.
Oh, a lot of things change with Benjamin Button, but I think I remember reading as you get older, how your brain waves react to anesthesia changes. And that's important because as you get older, you're more prone to undergoing surgery. So whether that means anesthesia is less effective on older people, does it have longer lasting side effects? Things like that might be a consequence of that. But brainwaves do seem to change because your brain is changing.
Topic.
So Vivian, what are you thinking about younger, and how would that affect what we've been talking about today?
Well, if the incoming input, so the V4 gamma oscillations exist before before V2. Like I'm talking about in the adult brain, the V2 input doesn't, at least as far as I can tell, doesn't have any role in generating the oscillatory activity in another brain region. So it's something that has to happen locally. And which is the brain wave is generated by, like Tim said, lots of neurons firing together, or turning on and off together. But I just, it seems a little bit like a chicken and egg situation, do babies have brainwaves? Maybe it differs. In terms of we know that certain parts of the nervous system develop first, are kind of going like this, or mature first. And then, because the prefrontal cortex, for example, matures much later. And in some of us, it is still not matured. I just think in whatever state brain waves are in children of different ages, like if we're talking 6 months, 6 months, a year, 18 months, 5, they probably are changing. The same frequencies are going to be there, but to what extent one is dominant or another, and in what brain regions, and coherence and synchrony between different brain regions might be more beneficial to a younger organism, but then lose their advantage in an older organism and something else takes over. I haven't read about this stuff in babies or in children, but I think it would be really interesting to dig into, to maybe make that young organism versus aging organism, and say what's the kind of arc of all of this? This is the middle piece. Well, with respect to attention, we're discussing the middle chunk, which is in a adult. But what about, where did we begin and where are we going? So that might be something interesting to revisit.
Yeah, especially since there's a lot of developmental things that can happen, ADHD and autism. Are there early biomarkers that you can detect based on brainwaves? I have no idea. I'm guessing no, because I think biomarkers for neuropsychiatric disease is really difficult. People have been looking for a while. So
the one last thing I was going to say, because I know we're running up to our deadline cutoff time, is that with respect to the younger brain, children are smaller, their brains are smaller, and there are studies, my goodness, that have been done with monkeys to map the time, literally the time it takes for an electrical signal to go from this part of the brain to that part of the brain. It's 350 milliseconds in a brain of this size. And so it's going to be shorter in a brain that is smaller. And as we are growing, our gross brain size is not going to change. If you're pregnant, or if you have Alzheimer's, well, not with Alzheimer's, because that's kind of a downward slope and it doesn't appear to, you can't go back up. But with pregnancy, you do see a kind of thinning of the cortex in certain places, and then a resprouting, and this, a depuffing and a puffing of the cortex because of changes in synapses and connections and the dendritic arbors. But like overall brain size doesn't really change, like kind of front to back, side, medial to lateral. But it does if you are, looking at an infant and then all the way up to, I don't know when children's brains reach their kind of like maximal size. But the time it's going to take for a signal to traverse one place to the other and to be processed at a very basic level, like, S1, like primary somatosensory cortex or primary motor cortex, primary visual cortex. So we're just talking about getting the stimuli in. And we're not even talking about like what you do with it, how it travels up to higher brain centers and then how that directs your behavior. That's going to happen at a different speed. What impact does that have on synchrony of different brain waves? And how is that going to influence the child's behavior? Like I try to keep in mind, this is the last thing I'll say, parenting tip from Vivian. Cut your kids, and I'll follow up and say like what age this is cut off, but cut your kids a little slack when you're like, pick up your shoes. There was like half a second between when you said pick up your shoes the first time and pick up your shoes the second time. I guarantee you that the information has not even made it to where it's supposed to go and then has not been processed, because also the stuff that we have in our brains that helps those signals go fast and go true to where they're supposed to be is myelin. It's that insulation, and children are still actively being myelinated. So it can't travel very quickly. It's going to take the time it's going to take. So cut them a little slack.
When do we have to stop doing that? What age roughly?
I don't know. I'll get back to you. But I have an 11-year-old.
If your undergrad's not picking up their shoes when you tell them to, then.
No, I'm thinking about adults because sometimes I have to say things. So maybe I say it too rapidly in succession.
Yeah, it could be. It's
turn on the microphone. Turn on the microphone.
And then each incoming wave of stimuli, suppresses the last one. And so you're just spinning your wheels. Yeah. So lots of interesting ideas and little thought experiments. And, there's some homework for listeners to go try and let us know what they think.
Cool stuff. Thank you, Vivian.
My pleasure.
It is TWiN64. You can find show notes at microbe.tv/TWiN. If you have any questions or comments, TWiN at microbe.tv. If you enjoy these programs where we give you science, real science, no lies, no disinformation, we'd love your support. Microbe.tv/contribute. Tim Chung is at New York University. Thank you, Tim.
Thanks, Vincent. And thanks, Vivian. That was a great opportunity to remember brainwaves and talk about it and learn something.
Remember brainwaves. Vivian Marsden's in New Orleans. Thank you, Vivian.
Yeah, my pleasure, guys. Thanks for all the help, Tim, because I also had to dig deep.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank, I have no one to thank. You've been listening to This Week. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.